So thank you again, Cuban, for this, for the invitation, for the nomination for the William Feindel Lecture. I must say I'm very touched. Dr. Feindel was quite a person, and I'm going to actually start by saying a few words about about Uncle Bill. Now, Bill Feindel did not particularly like, I think, the term Uncle Bill, but amongst us who were considered, like we considered him our uncle, in the sense that he was the uncle that made sure that everyone in the family was well taken care of. And I'll tell you about how that is. So Dr. Feindel was an incredible surgeon, professor, researcher, and mentor. He was just exceptional. He was a neurosurgical pioneer. He trained and worked with the best. So here, let's go back here. Now I have to figure out how to get my, yes, we have a nice big cursor. So trained with the best. Jesus. All right, sorry. I'm usually not this nervous giving a talk, but it has been 2 years. So here he is with Wilder Penfield, and here with Ted Rasmussen, and of course that's Dr. Feindel there. He recognized very early on how technology could help out in the lab, and I can see that this pointer thing is not going to work at all. So I'm just going to point to the screen, but then that's not going to really work. So here we have an example of the Montreal procedure that was developed by Penfield, where they did electrophysiological recording and stimulation on the cortex to map out the brain during surgery in an awake patient. I'll just let that sink in so you can think of what that means in terms of surgical procedure, anesthesiology, nursing, all the staff around, and think about the patient. So that's all just blows my mind. So as Christine said, Dr. Feindel was instrumental in bringing imaging into the neuro. So here we have the very first CT scanner that was installed in Canada, an EMI scanner. This is the same EMI company that produced the Beatles records, just so you know. He also was instrumental in making sure that we had the first PET scanner, the positome, and he also made sure that we had the first superconducting magnet in Canada. And in gathering all of this equipment for imaging, he essentially founded the Brain Imaging Center, and he was the first director of the Brain Imaging Center. He was also an avid medical historian. He was quite fascinated by medical history. He was quite fascinated by how one learned over time about the body. He edited the tricentenary edition of Willis's Latin treatise, Cerebri Anatomae, which already he edited the Latin version. Just so you know. Okay. And he was a great mentor. And this is the part. So he was absolutely fantastic. So when I was a master's student, I worked on techniques for image-guided surgery with Terry Peters. And we did volume rendering and stuff for surgical planning. And we had these really nice computers that you would program, and you could get some stuff to build an image, and then you'd run the program, and then 35, 40 minutes later, you might get a single frame that you could look at and go, whoa, I made a mistake, I got to do it again, and then go for another coffee. So Dr. Feindel needed some 3D figures to show the hippocampus for a course that he was given, and he heard that we could do this kind of stuff. So he came down the lab, sat with me, worked with the segmentation programs, taught me where the hippocampus was. Me, computer science, hard-headed, didn't want to know anything about anatomy, but he taught me anyway. And we used our tools to segment the hippocampus, and then we produced the 3D images, which he then used for class. So that was a really nice afternoon. You know, I was spending my time with Dr. Feindel, one of the greats of the MNI. After that point, it was incredible. Every time I saw him in the hall, he would ask me how my project was going, how my master's was going. Did this experiment succeed? Did I think about doing that? Was I going to do this, or was I going to do that? He knew what I was doing all the time, which in one sense is kind of scary, because if you're not advancing, he knows it. But he had this interest in everybody that was at the M&I, which was quite spectacular, because at the time we were like 300 people at the M&I, and half of them were researchers, and he knew what everybody was doing. He knew what the floor cleaners' kids were doing in school. The guy was absolutely incredible. He was truly interested in everyone, and he wanted the best for everyone. So, as my voice cracks up, sorry, with this in mind, it is my great pleasure and my honor to give this talk. Give me just a sec, maybe a little glass of water. Sorry about that. So I'm going to show you some of the research that we've done in the lab with by the students in my lab. And if ever I say we did something in the lab, it's not we. It's the students, it's the postdocs, it's the fellows. I write the grants, I write the progress reports. They do the interesting work, and they leave me in the office to suffer with the paperwork. I also want to give you an idea of what happens to graduate students when they finish. So this is kind of the, hopefully, mentorship role of this talk. So let's start quickly. So radiological images were used up until about the 1970s for surgeons to figure out what was in the body when they were trying to plan surgery and trying to figure out what's going on. So we have 2 skull x-rays here. Of course, in the late 70s, we get CT. That's going to give 3-dimensional slices through the body where you can see bone and soft tissue. We have MRI that's going to show you exquisite soft tissue detail. We have positron emission tomography, which is going to be able to show you the function of the tissue. By doing some fancy image processing, registration of the data, you can put the PET on top of the MRI, and you can see what part of the anatomy is actually functioning. For all of you, this is bread and butter. 25 years ago when we started, this was cutting edge. Of course, with MRI, you can do functional and magnetic resonance imaging. You can use the functional magnetic resonance to show the function, again, overlaid with the anatomy. You can use digital subtraction angiograms to look at the vasculature in the brain and other structures of the body. And of course, we can do this now with MRA, magnetic resonance angiograms as well. And of course, we can use ultrasound that can also show tissue and vasculature, a bit as Hassan showed earlier this morning. So in the NIST lab, a core group of my students developed image processing tools to segment, tissue classify, extract cortical surfaces, register data, do nonlinear registration, segment the different structures. So that's a core group. Half of my group uses these techniques to analyze data from patients with neurological disease and see how patients with disease differ from normal controls and how these patients, their brains change with treatment, pharmaceutical or other. And of course, I'm not going to talk about any of that today because it's going to be about image-guided surgery. So if you want to hear about that, you'll have to invite me back for, I don't know, Bill Feindel's talk too. So there we go. So I'm going to give you a whirlwind tour of image processing to plan surgery, and then once planned to help guide the surgeon during surgery. And a bit of an overview of the talk. I'm going to start with some of the planning that we do for atlasing and Parkinson's in order to plan out the surgical procedure. A little bit of basics about image-guided surgery in general, what's needed to do so. And then I'm going to talk about the platform that we've developed. Again, notice the we. I'd like to take some part of ownership, but it's really, I'll tell you who did it in a few minutes. And 3 applications of our IBIS platform. And then I'll end, hopefully, with what we've learned about image-guided surgery and then what I've learned about supervising students. So Parkinson's disease is the second most frequent neurological disease after Alzheimer's disease. It's caused by a reduction in the dopamine neurons in the substantia nigra, and it causes symptoms like resting tremor, plastic rigidity, bradykinesia, and it affects about 1 in 100 Canadians, which is actually a reasonable number of people. Of course, pharmaceutical treatment can be done at the beginning of the disease to help minimize the symptoms, but after a while, for quite a number of patients, the pharmaceuticals no longer work and they become candidates for surgery. And the surgery is to place an electrode deep in the brain to modulate the networks and the pathways that are affecting the symptoms. And in order to do this precisely, because the nuclei you have to target in the brain are very small, like the subthalamic nucleus is the size of roughly a watermelon seed. Or let's say even better, a cantaloupe seed, because the cantaloupe is the size of the head. Now imagine going with a needle from the outside of the brain into the cantaloupe to extract or just to touch one specific seed in the cantaloupe. That's stereotactic surgery. And that can be done with these stereotactic frames. And what they have are these fiducial markers that are on the side of the frame that we can see in the images. So if you could identify the anatomy in an XYZ coordinate by moving these tool carriers on the side of the frame or the angular arc, you can attack or you can attain any point with very high accuracy during surgery. And this has been the basis for stereotactic neurosurgery since the 1950s. And it's been quite fantastic. And so we'll go to one of the first students that was in my lab, Malar Chakravarti, who was sitting around here someplace. So he was here from 2001, 2004. And his goal of his project was to be able to identify some of the targets that cannot be visualized on the MRI. And so to do this, he took a bunch of histological images that were here, where these contours were actually drawn on photographs by the neurosurgeons to identify the different structures that they could see on these histological photographs. He stacked all the images, well, digitized the images, stacked them all together, aligned them all together to build a 3D atlas. There we go, so I can point to that. A 3D atlas of the, oh, isn't that great? I'm so sorry, there's another monitor here that I can actually see instead of always looking over my shoulder. So there we go. So there's this nice, he built this beautiful 3D model of the deep basal ganglia and the deep gray nuclei, of which many are targets for imaging, for stereotactic surgery. And so he then used this data to build a pseudo MRI, so essentially colored in these regions with the MRI intensities that are similar to an MRI data set like that, and use that to drive an image-to-image registration so that you could align the atlas here with the MRI. And then once you had that, you had a template and an atlas aligned together such that you could use any new data, a new patient data, you could take their MRI data, align it to this template, and then you could take the labels from the template, push them back onto the subject, and you can identify the nuclei that you couldn't see on the patient's MRI, thus identifying the surgical targets that are necessary to be treated using the stereotactic neurosurgery. So that was fantastic. And so that's all the notes that I had. So there we go. After Malar finished off and has gone off to build an incredibly nice career, which I'll talk about in a few seconds, another student in the lab, Yiming Xiao, put together an imaging sequence such that we could actually see some of these deep nuclei. So when Malar was doing the work, we had this column 27 average. We mapped his atlas onto the column 27. Column 27 was 27 scans, but he was about 27 years old. That's quite a bit younger than most Parkinson's patients. And so Xiao built this 10-echo 3D flash imaging sequence that gives simultaneously a T1, a T2, a phase, a SWE, and an R2 star map, all in 7 minutes with just better than 1 millimeter isotropic resolution. So this was quite fantastic. And here we can see the zoomed in regions here, where just here you can make out the right nucleus, the subthalamic nucleus, and the substantia nigra. So one of the other issues that we had with the initial template, it was created by the anatomy from a single subject. And while it was a single normal subject, at least we hope Colin was normal, he was a postdoc in the lab at the same time when I was in the Brain Imaging Center. His anatomy could be a bit of the extreme of the population. So Xiao went and grabbed 15 PD patients, their MRI data, ran the multi-echo flash on them, and then built an unbiased template average of those subjects using this group-rise registration technique that was designed by one of the RAs in the lab, Vladimir Fonov. Now, once that's done, we have these templates that you can see here that have beautiful contrast, incredible cortical detail in the T1, fantastic detail of the deep nuclei in the T2. These two images can be merged together in such a way, by taking into account the phase map, they can be merged together so you can get the detail of the deep nuclei in the center of the brain where you want it, and keep all the cortical detail that you're going to need eventually if you want to use this for surgical guidance in surgery. All of that in the same image, in this fusion image. Here's a zoomed-in view of the 5 templates where we can see the fusion image, the T1, the T2, again the R2, and the phase map, and you can really see fantastic detail of the deep nuclei. And so, because now we have additional contrast in the deep brain, we can use that additional contrast to improve the registration of a single subject, PD patient, onto the template so that we can bring the atlas structures from the atlas that are aligned with the template onto the patient, thus achieving better accuracy for stereotactic surgery. And so Xiao went and put the atlas, doing the same type of registration technique that Millar had pioneered 8 years before, and aligned the atlas onto the templates so that we ended up with this great identification of the basal ganglia structures and the deep nuclei that you can see here. And so the question that students often ask is, well, what do you get once you get a PhD? What happens to you? So, what happens to Millar? He becomes an associate professor at McGill, at the Douglas. He's director of the Cerebral Imaging Center and director of neuroinformatics. Yiming Xiao is now assistant professor in the Department of Computer Science at Concordia. And while Millar doesn't do a lot of image-guided work, he does do some DBS, some deep brain stimulation in mice. So if you think you have to be accurate when you're looking at a brain this big, imagine how accurate you have to be when you're looking at a brain that's that big. And Xiao continues to do deep learning landmark detection, still works on Parkinson's templates and atlases. Our group has also developed a number of sophisticated image processing techniques to identify specific structures. And in this case, we can see blood vessels in 3D from CT angiography. We've also developed techniques to extract the cortex, so the cortex and the cell side that are on the cortex and the blood vessels can be used as landmarks that the surgeon can see when they've done this craniotomy for their surgery so they can align themselves with the images. Now, this brings up one of the important issues in surgery. For the images that I've shown you up until now, they are pretty. In fact, I really quite enjoy them. I think that the whole idea of this image art, I think these images are quite beautiful. But they are only pictures of the relevant anatomy. The surgeon has to use subjective knowledge to go from the images on the screen to the subject. It's not like we have a coordinate system that was given to us by a stereotactic frame when you're just doing open surgery. And so what we want to do is to achieve the same thing that we had with a stereotactic frame, i.e. a coordinate system to identify the structures, a coordinate system to bring tools to those targets. We want to do that, but without a frame. And this is called frameless navigation. Like a GPS navigation system in your car that allows you to plan your trip and to guide you step by step of the way as you get there. In frameless navigation, we want to do the same thing. We want to do the image to patient mapping, and we want to have accurate tool guidance. And all of the tools that are listed up, all the goals that are listed up here have driven the research in my lab over the last 15 to 20 years. So in neuronavigation, we need 3 things. We need here a camera that's going to track the patient and the surgical tools in 3D space. We need to be able to actually track the tools, and the tools have to have these little markers on them so that we can actually easily see inside by the camera. And we need to have some type of computer system to be able to display the results. So to achieve the patient to image registration, points on the surface of the patient are identified with a track pointer, as you see here, and they're identified on the 3D rendering of the surface of the computer images. And we identify the homologous points. You can do a simple least squares transformation to figure out the transformation between the patient and the images. And of course, the inverse transformation of the images to the patient. Once this is completed, the surgeon can point to any location on the anatomy and see that location in the images. Or you can virtually extend the tip of that pointer by 4 or 5 centimeters to see what is 4 or 5 centimeters deeper past the end of the pointer. That's giving you kind of a Superman vision. You can see below the surface. And I'll get to that a bit later. This video also shows one of the difficulties of the current commercial systems. The surgeon has to look away from the patient to the computer screen to be able to see that information. And we want to try to address that. So 2 software engineers in my lab, Simon Drouin and Anka Kochanowska, developed our neuronavigation platform known as IBIS. And this has been done over the last 15 to 18 years. So IBIS is a publicly available open source neuronavigation system, has all the functionality of a commercial system, and because we have all the source code, we can tweak it, use it, develop it any way that we want. It's been very nicely written to have to be easily extensible with kind of an app add-on. And so we can develop new techniques in the lab, test them in the lab, and then have the surgeons look at that. And if they like it and we have the REB approval, we can actually bring it into the OR. And this we can do from like concept to the OR. We can do that for simple ideas, 3, 4, 5, 6 weeks. Whereas if we were working with a commercial system and doing that would take 2 to 3 years. So in terms of development, it's fantastic. Here's the IBIS system in the surgical suite. You can see that we're quite cramped in the surgeries at the M&I. So in red is the Medtronic FDA-approved commercial system, and in blue is our system. We use our own camera to track the tools and the reference, but we're using the same tools and the same reference as the Medtronic commercial system. This way we don't modify the current standard OR procedure at all. We're just stealing the video. We're not even stealing the video. We're acquiring a second video that we're going to use for our own processing. So the IBIS navigation system can show multiple modalities in 3D objects all simultaneously. Here's the data from a 36-year-old patient with a glioma, a right frontal glioma in pink. We've got DTI fiber tracts in yellow. We have fMRI here in spectral colors. And we have in purple here are blood vessels that have been extracted from a gadolinium-enhanced image. In the lab, our neuronavigation system has sub-millimeter accuracy, which means that we can point to some location on a plastic brain or a plastic skull and see the corresponding location within the images to within 1 millimeter. So the surgeon can be quite confident that what they see in the images is what they have below the surface of what they see on the patient. But in reality, in the operating room, the systems don't have sub-millimeter accuracy. So this list shows errors of 2 to 12 millimeters are reported in the literature. And there's a number of causes that have been identified in the literature. They go from errors in the initial point-to-point registration that's done on the skin, geometric distortion in MR images, but there's also very important biological changes that happen during surgery. And that's what I'm going to talk about for the next bits of the talk. So this is the best figure I found in the literature, and it's now close to 20, well, that's actually over 20 years old now, but it's one of the best ones. So this is acquired by Arya Nabavi, neurosurgeon at Harvard, Brigham and Women's at Harvard. They had a double donut MRI system, intraoperative MRI system, which is quite nice. So this first image here, there we go. The first image shows an MRI image like many of you have seen and work with all the time, right? Everything's intact. At least I hope all of your patients or all of your subjects have intact skin and skull and brain. But after the surgery starts, of course, you remove the skin. So here you can see a bit of the skin flap. The skull is removed, the dura is opened, and the brain begins to swell. And you can see that here with the brain actually pushing out because the heightened intracranial pressure is pushing out of the actual craniotomy. The surgeon says there's too much intracranial pressure. The anesthesiologist plays with mannitol levels, oxygenation levels, and the brain will sink down a bit more and then become safe again. During the surgery, of course, the surgeon is going to be active doing something like a resection, and this is showing the resection cavity. And you can see in this resection, which is quite deep, that the brain is actually shifted down from where it was. And here you can see it's shifted down away from the inner table of the skull. And this shift from the lowest point to the highest point here could be on the order of 4 to 5 centimeters, not millimeters, centimeters. So we have an engineering solution in the lab that's good to sub-millimeter accuracy. Biology throws a wrench in the works where I think the technical term is screwed. We could use MRI images, like an intraoperative MRI as they do in Harvard, to get new images each time the surgeon thought, the neuronavigation system is a bit off with the images that I'm using. But this is expensive, and that requires, expensive, very expensive. Requires magnet-friendly surgical tools. Those are expensive as well. And the anesthesiologist has more difficult work because the tubes that go to the patient have to be longer because you have to be in the surgical area, and then you have to be able to move the patient into the scanner. And all of that, of course, takes time. And this is the biggest issue with intraoperative MRI for surgery, is that it takes 15 to 20, 30 minutes to prepare the patient for imaging. Then you have to take 10 to 15 minutes of imaging and then 5 minutes to bring the patient back. So between the time the surgeon says, the images are not terribly precise, to the time that they have new images, it could be almost an hour. And for neurosurgery, you don't want to spend 3 sessions of imaging during a surgery. 3 hours is just completely unacceptable. So we want something faster. Ultrasound is an imaging modality that can be used to do this. Hassan showed some really nice images this morning that he acquired as a postdoc He acquired with the neurosurgeon. The neurosurgeon was always holding the probe on the patient, just to be clear, in case anybody's here from the REB board. So there's many advantages in ultrasound. It's very inexpensive, orders of magnitude less expensive than intraoperative MRI. It's intuitive. It's real time, as Hassan showed this morning. There's no need to move the patient. There's no need to do a lot of prep in the patient. Essentially, images, we need new images, get the ultrasound probe, fill the saline, do some imaging, give the probe back to the scrub nurse, that's it. There are not 100% good things. There's a few difficulties with intraoperative imaging. There's a learning curve for the surgeons. They have to learn how to interpret the images. There's a limited field of view. We don't see the entire brain at one time. You only see a small conic region. And the images can be difficult to interpret. And so if I come back to IBIS, in terms of its extendability, IBIS was extended to be able to use it to acquire ultrasound images during surgery, and also to show data with augmented reality, which I'll show in a few slides. So I'm going to go back into the OR and show you what the OR setup looks like. So here we have the IBIS workstation with a tracking camera. We have the ultrasound machine. We have the surgeon that's here holding a tracked pointer, and here a tracked ultrasound probe. Here's what's called a dynamic reference object. This is what gives us the coordinate system tracked by the camera that defines the origin of the whole space. And so the big advantage here is that the ultrasound is going to represent the physical reality of the tissue at that point during surgery. And acquisition is quick, no patient prep. You can essentially do this as often as needed in surgery. And recall the issue that I spoke about, this fact of having to look away and then look back on your patient, going back and forth. We've got Simon, or Simon, I should say, and others have worked on augmented reality. So we actually inject computer models of the tumor or of blood vessels or other objects of interest into the operating microscope. So you can actually see, now with real Superman vision, here a nice green tumor. Tumors are not green. And they're green in cartoons, they're not green in real life. I can't remember what this little blue area is representing, but tumors are not cyan blue either. So this gives us the potential with ultrasound correction to correct for brain shift, and with augmented reality to be able to see the corrected data in the OR. And so the next sets of slides are all to show that, or to demonstrate that. So with 3D ultrasound is very similar to MRI, but at the same time there's some significant differences. And here you can see the same tumor image with both MR, and the ultrasound. And the ultrasound can be somewhat difficult to interpret, although here it's been reoriented with the MRI. You see the MRI, you see the MRI features, you can identify the longitudinal faults, and you can carry that information off to the ultrasound, and you know what the ultrasound. But if I showed you just the ultrasound, maybe not. So the question is, can we use the ultrasound to correct the preoperative MRI? And that has driven a ton of research that I'm going to show over the next bit. So, please, there we go. So Tal Arbol was my first, actually was my second postdoc in the lab. And so she was there from 2000 to 2001, and she developed a technique to build a pseudo ultrasound. And the idea with this is to take the original MRI, do a segmentation, compute some gradients, add these like a color-coded intensities from the segmented MRI with the gradients to build this pseudo ultrasound. The pseudo ultrasound could then be matched to the MRI just using standard cross correlation. So a really simple similarity metric, and that would work. So here we can see in the center image here, in blue is the ultrasound showing the physical reality of the surgery at that point. MRI in gray is showing is the preoperative MRI. But you can see here easily that there's at least a centimeter offset. So the brain has shifted by 1 centimeter. And so the surgeon can't trust the standard commercial neuronavigation system. And in fact, the surgeons use the navigation system to plan the craniotomy. They use to plan the corridor after they've opened the dura. And then they push it off into the corner and they don't use it again because the brains move. So we continue this work with Laurence Mercier, who is another postdoc, who's a PhD student in the lab from 2004 to 2011. And you might think that's a long PhD. She had 2 kids at the same time. And this is one thing that I think is really important as a supervisor. You have to support your women students. Absolutely key. So in this, Laurence went and augmented the pseudo ultrasound technique, doing a bit more segmentation of additional structures, modifying the technique to get a slightly better pseudo ultrasound object that we could then use to register with the MRI. She then went and compared that to a normalized mutual information technique for registration that was the standard similarity function that was used. And she found that in every case that she tested, the 15 cases that she tested here, that the pseudo ultrasound was either better or equal to the normalized mutual information, and that she managed to bring the misregistration down to about 3 millimeters. So we're not sub-millimeter as we are in the lab, but we're much better than we were before going. So, and the other big advantage of the technique that she developed was instead of the 20-minute procedure that was required when Tal was working, Laurence's procedure took about 3 minutes to reconstruct the ultrasound volume and 5-6 minutes to compute the registration and resample the MRI. So we're down to under 10 minutes from the previous 20. Ingrid Reinerson was another PhD student in the lab. She too had 1 or 2 kids while she was doing her PhD. So she was either very efficient with her PhD or very, very efficient with her PhD, depending on the number of kids. I can't remember. She started her PhD looking at vessels and could vessels be used to drive the registration. In order to test this, she built a phantom. So this is made out of PVA, so it's polyvinyl alcohol cryogel, essentially a starch that you mix with water, you freeze and thaw, and when it's thawed, the object, which we can see here, has the mechanical properties that are very similar to brain. So here we used a novelty mold to get the kind of a sulci brain shape. Used a Foley catheter underneath so we could inflate the catheter and deform the brain to simulate brain shift. And this is just showing a 3D rendering. So when she used this and took very, very careful measurements of landmarks on the surface, she showed that she could get much better than sub-millimeter accuracy on the identification of landmarks. And so that meant that she could use this phantom to see how well the registration techniques would work. So she developed a registration technique used a Frangi vesselness filter to extract the blood vessels from MRI and from ultrasound, used a modified and iterative closest points algorithm to make it more robust, and she did a two-step registration process, starting with an affine registration, get the initial guess, and then doing a nonlinear thin plate spline afterwards to finish the job. So here on phantoms, she got great results. So here you can see after linear registration, almost all the curves, the target points and the source points, the green and blue, are almost all aligned, except for some places here where there's this nonlinear deformation, a nonlinear deformation that can't be accounted for using just an affine transformation for the registration. But if you apply the thin plate spline afterwards, that gets fixed. And so that worked out really well. When applied to clinical data, she also obtained very, very good results. So this is some images or a cropped volume of the middle cerebral artery. And this is just to show you, give you an idea of where the middle cerebral artery is from a top view of the head. Here in red are the MR vessels, in blue are the ultrasound vessels. And so before registration, we're almost a centimeter off, very similar to the other images that I've shown you where it's roughly a centimeter off. After linear registration, better than a millimeter accuracy, so it's a tenfold reduction in error. And and after nonlinear, it gets a little bit better. But this just goes to show that while there are some regions that do align better, on average, there's not a huge improvement. But this essentially shows that for some cases, linear or affine transformations might be good enough for guidance. This is something that still has to be evaluated. In the paper that she published, she looked at data sets from 5 different clinical data sets and showed that in every case, we could get sub-millimeter accuracy on blood vessels. So that was great. Another PhD student in the lab, Sean Shen, that came after Ingrid, continued to work on the same project, but started with looking at the PVA phantom, but here improved the phantom by using the column 27 brain, so this three-dimensional template brain, extracted the cortical surface from that, used that 3D surface model to drive a 3D printer to build a 3D flexible mold that has a nice deep sulci and a complete insular region. So it's not the little toy model that we had before. It's much better. And had also built a lateral ventricle as well. And so here you can see the phantom that we have. He too did the other thing that he had improved for the phantom was to make it multimodality. So these first 6 images are from MRI. This is CT, if memory serves, and this is from ultrasound. And here's just another picture of the phantom. So the registration technique that he used was very similar to that from Ingrid, although instead of just using the Frangi vesselness, he combined that with the original intensities. Also did a 2-step registration, but modified it by starting with a grid transform registration and then refined it with blood vessels. Not that it's terribly important, but he got reasonably good results. So in the end, when evaluated with landmarks that were not on the blood vessels, but were on landmarks that were spread across the surface of the cortex, he did in hundreds of simulations. Where's my mouse? Sorry. He found that he could get better than 2.5 millimeter accuracy almost all the time, even when the initial transformation or misregistration was quite large, up to 2 centimeters away. And so another postdoc that was in the lab, a very productive postdoc, I must say, there from 2010 to 2014, was Hassan Rivaz, who you heard this morning. So in the previous work with Tal and Laurence, they built these pseudo-ultrasound data sets to try to get around the fact that there was ultrasound and MRI are difficult to compare to each other. And then Ingrid and Sean also tried to get around that by extracting blood vessels. Hassan said, none of that. I'm taking the problem head on. I'm going to take ultrasound. I'm going to match it to MRI. I'm going to figure out how to do it. And he did so 3 different ways. He looked at 3 different statistical techniques. The first one was self-similarity weighted mutual information. The second was contextual conditional mutual information. And the third was a patch-based correlation method. Each one of these techniques used different statistical techniques to compare local patches of the tissue between ultrasound and MRI and use that to compute the similarity locally so that you could drive a nonlinear registration that I believe was driven by B-splines, if memory serves. And in each case, Hassan got great results looking at landmarks that were spread across the volume of better than 2.5 millimeter accuracy. So once again, bringing data that was misregistered back into registration so that the surgeons could use it. And where are they now? Can this actually be read from there? I hope so. So Tal, Ingrid, and Hassan all obtained academic positions. So Tal is a prof in the Department of ECE at McGill. Ingrid is an adjunct professor in Norway. Hassan is an associate professor in computer science in Concordia. I missed two of them, unfortunately. Well, unfortunately, not unfortunately. I take that back. Sean went to industry because he wanted to go to industry. He did not want to write grants for a living. He saw me losing my hair, saw me going white with my hair. Thought, nah, not for me. So he decided to go to industry, where he worked for Synaptive Medical in Toronto, and more recently for Moleculite. And one other little small parenthesis, although I'm running out of time, for Sean is he edited 2 books on Chinese gastronomy recently and published them. I think that that's pretty cool. It's like, find what you like and do it. That's it, it's just good. Laurence left academia as well. She gives courses and conferences on meditation training, which maybe I should follow. Okay, I'm going to come back to Simon Drouin and Anka that developed this, our IBIS tool. At some point, I don't know if I think I was serious when I told it to Simon, but I made him an offer. I said, you're doing really great work. It's really cool, the stuff that you're doing. You could probably get a PhD out of that. But, you'd have to do the same work. You're going to have to write a bunch of papers in addition to it. And I'm going to cut your salary by half. Surprisingly, Simon said yes. So he started, for this PhD, after developing tons of stuff for IBIS, he started by developing Prism, which is an interactive design for GPU volume rendering shaders. So essentially, by writing a few lines of code, you could actually build a specialized volume rendering, driving the shaders on the GPU, the graphics processing unit. And because GPUs have become so powerful with all the gaming, it's become spectacularly used for volume data. And so here you can see an example of volume carving. And the volume carving, I find this just gorgeous. You can see the interior of the brain. You can see the interior structures. This is fantastic. What's even more impressive? That's it. That's all the code that had to be written to do that. It's like 9 lines. So volume shaders are really powerful, and especially if you can build the interface where you can tap their power with very little effort on your part, it's fantastic. Simon continued to use the volume shaders as he was very interested in developing new and more efficient visualization techniques to drive the visualizations. He looked at static and dynamic shading, thinking that dynamic shading would be able to give you a better percept of depth. And for static shading, you just have the light in one place and you look at the object, nothing moves. In dynamic, you get to move the lighting around. So here, imagine a lantern put at the end of the pointer, and you move the pointer around in the object. And here you see, as the lighting gets changed, this is like wandering around in the woods and lighting up different branches with a lantern. It's quite beautiful. He also worked on augmented reality visualization. So this is where we're really getting into the meat and potatoes of the new visualization techniques for image-guided surgery. So here we can see in color blood vessels that were extracted from CT angiography, overlaid on a plastic phantom of a patient that had an arteriovenous malformation. And so as you move the pointer around, you can see what is in the neighborhood of that pointer below the pointer. So this is quite nice. And I don't know if we'll be able to see at some point the video. It actually shows the AVM, but we'll skip that. Before the pandemic, we used the IBIS system relatively often in surgery. So when we first started, we had to beg the surgeons, and they were really patient with us and very nice, allowing us to gather data in surgery. Now they actually come down to the lab and they'll ask my students, we've got this complex case, small tumor deep in the brain. We might miss it with the standard navigation. Can you guys come up and just drive it with the ultrasound? So here, this is showing what augmented reality can do in one step of the surgery. So this is defining the craniotomy. So in the upper corner here, this is what the surgeon sees when looking directly at the patient. So that's the surface of the skull. Here is the augmented reality view. So here we had an iPad that was just next to the patient. The iPad was tracked, looking at the image, projecting the position of the tumor quite accurately, allowing the surgeon to go dot, 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 dot. If I dot here, I'm going to make the most tailored, most appropriate craniotomy for this subject. And then it's just a question of connecting the dots, as if neurosurgery is so simple. Marta Kirsten was another PhD student in the lab that worked closely with Simon on augmented reality. She was very interested in the human-machine interface aspects of the navigation platform. And in particular, she was interested in evaluating the different types of rendering systems that we could use to see what would lead to the best for vascular surgery. And so here she did a task where the task was presenting the images like this with different renderings and tried to decide between 2 points that were on different vessels, which one was the deepest. This was run with non-experts, i.e. non-surgeons, so people in the lab and surgical residents. The chroma technique, which is this pseudo-chroma depth here, was found to give the best results, the best accuracy in deciding front and back, so the depth percept. And when done with surgeons, we got the same result. One thing that was really surprising was the edge description here, the edge enhancement, which is shown here, was actually second best and almost as good as chroma for the surgeons. And we interpret this as the fact that surgeons, unlike the non-experts, actually have a ton of anatomical knowledge. So when they see a little vessel that does this, they know which one it is, and they know that that one is actually in front of this one that's nice and straight and goes that way. So Martha went and looked at multiple surgeries and went to go see where the techniques were useful. So at the very beginning, augmented reality was good to plan the craniotomy on the dura. And once the dura has been opened and on the cortex, augmented reality was very good to plan the resection corridor. And at the end, or not at the end, but once you actually corridor down to the target, it was very good to decide what to do next. So for AVM, it's very important to clip the feeder arteries before clipping the draining veins. If not, if you clip a vein, the AVM will continue to receive blood and then increase in pressure and perhaps burst. Not a good thing if you're a patient. Yes. So right now I'm at 46 minutes. I have, unfortunately, another 22 minutes to go. No, I have a bunch more slides. There are some that I would really like to show, but I think I could just skip this and jump to the end. What do you think? Or do you want to give me another 5 minutes? Yeah? 5 minutes? Okay. All right. So Ian Girard was another PhD student in the lab. He's about doing his PhD at the same time as Simon and with Marta, and he was really interested in looking at augmented reality and brain shift combined, correction of brain shift combined, in tumor surgery to see how well that would work out. So he started by looking at accuracy in surgery, built out this linear test apparatus, which I think I'm going to skip because it's not that interesting. What is really interesting is the augmented reality work. So here you can see Dr. Hall occurring ultrasound on a patient. Here you can see the actual MRI of that showing, once again, it's about a centimeter off. So in the gold colors is the ultrasound, gray is the MRI. And so you can see that there's this misregistration, and you can understand why the surgeons don't like to use a navigation system once they've gotten into surgery, into the resection. And so here, we can actually correct for that using all these different registration techniques. So here, the registration technique that I'm showing here was by one of Tal Arbel's PhD students. So this is one of my academic grandchildren, if you will. So he developed a technique and coded it on the GPU, thus getting pretty quick. So the image that I show you here, maybe I just show it one more time because it's kind of fun. So this fit that I'm showing is not done real time. That's not the real time. We actually have to slow it down, put it in demo mode, because when it actually works, it's 0.4 seconds. It just goes and locks on. It's really fast. And so with that, there's no reason not to use this. There's no delays. By the time that the surgeon does the ultrasound sweep, hands the probe back to the scrub nurse, everything is ready to be able to visualize and go again. Ian went and did validation with this system, and just to show one of the validations he did was he told the surgeon to put the pointer on the edge of the tumor. In surgery, what they saw is the tumor in surgery. And here you can see in the middle image here, before correction, you can see that the end of the pointer is actually in the tumor. Well, this is in the rendered tumor, given the preoperative image. But after correction, you can see that the edge of the tumor and the tip of the pointer are nicely aligned. So the correction is working. And of course, the augmented reality visualization is not bad either. So a quick where they are. Simon, prof. Marta Kirsten is a prof in Girard, not a prof. He went to med school immediately after his PhD. In fact, he delayed the end of his PhD so he could go to med school because he was accepted. So I'm going to go skip the work that we've done in spine surgery, which, while I think it's really, I needed my mouse, please. No. There it is. So I want to go to the part, what we have learned. So this is the take-home messages. So for image-guided surgery, you can imagine what we've learned. Essentially that neuronavigation must account for brain shift or for tissue movement in order to maintain accuracy. Otherwise, navigation's worthless. Multiple registration techniques can be used. More often than not, the registration technique may be tailored to the specific type of surgery in the specific anatomical location. And I think that we have speed that's necessary. I don't think we have to get faster than we are now. I think we could probably still improve on the accuracy because we're on the order of 2 millimeters, but even though I like to tease the surgeons, it's not rocket science where if you're off by, microns, your space shuttle explodes. It's only brain surgery. Augmented reality is useful in surgery, in fact, very useful because the surgeons actually like it a lot in order to plan different steps of their surgery, so this is really good. We still have to work to improve the visualization, and in particular to improve the depth perception, because there are sometimes when we do the rendering looks like it's on top of the cortex, even though we know the vessels are beneath. And the most important part is we've developed all these tools. The tools are great. All the students have gotten their PhDs. That's really good. But we need to evaluate the clinical efficiency and the usefulness of these tools. In every grant I write, we will diminish morbidity and mortality. Can we actually demonstrate and prove that the patients benefit? That's going to be the next step. For graduate students, and this is probably the part that might be most interesting for the graduate students, although I hope that you like the image-guided surgery bits. So for me, I've learned that if you build a good environment, a safe environment, students are going to do excellent work. There's just no doubt about it. Students are really brilliant. They will do excellent work. As a grad student, it's your responsibility to find out who's got a good lab, who is supportive. Go to those labs. That's really important. As I say, students are brilliant. They keep me on my toes. I get to learn stuff forever. I'm going to go to the grave learning new techniques. It's going to be fantastic. You need to ensure that the students feel safe so they can try new things so that they're not afraid to fail. And if they do, they're going to learn a lot. And of course, I'm going to learn a lot because I actually suggested that they're doing the stuff that they're doing. And if it failed, then I've got something wrong. So that's important. So we can learn a ton from the mistakes. And finally, while an academic position is hard, and I know that at these times, people that are looking for academic positions, it can be really hard. There's tons of competition. It's not easy. So, and of course, the call to industry is strong, and there's lots of money there. But that said, it's hard. It's incredibly satisfying. Seeing students succeed makes me incredibly proud. So for me, it's like I want students just because I'm egotistical. I want to feel that feeling again. So I'm going to end with two things. First, I'm going to thank all the grad students, the postdocs, and the research associates that have come through my lab. This is the list. There's, I think, 79 people. Those in the dark black are those that have worked on image-guided surgery. So you can see, if you want to hear about everybody that's in gray, invite me back. And the last thing that I want to end with, for this Feindel talk, is sharing this photograph of Dr. Feindel and Dr. Milner at their birthday lunch at the MNI a few years ago, on the occasion of the 95th birthday. Dr. Milner, when she was 90, thought that she would be, well, finally retiring, leave some space for the more junior profs. She got this, I forgot what it was called, an Italian equivalent of the Nobel Prize. She got $1 million with the only caveat, got to work with young people. So she had cleared out her lab. Oh, no, okay, you, and you. Come back to the lab. We're starting up again. And so she's been going strong. She's 103, 104, still goes to the MNI, still is working. Dr. Feindel came to the MNI almost until just a few months before he died, before he passed away. He always had a little black notebook in his pocket. When he would listen, he'd go to all the talks, he'd take out his notebook, he'd write off a few little notes. The talk would end. If nobody had asked a question, he didn't want the speaker to feel bad. He'd take out I have a question. You know, your work that you just described reminds me of a surgery we did, a surgical experiment we did in 1952. And then he would go on and describe, something that had happened. So that is the way I want to be on the end. My wife doesn't think that I should work until 95. She wants me to retire at 65 and do something else. But anyway, I want you all to think about what you like. What is your passion? What do you want to do until you're 95? And do it. Just do it. I'll end with that. Thank you.